NAS-G spt5 factors bind to their respective RNA polymerases at the tip of the clamp, overlapping the region contacted by the sigma factor 4 in case of bacteria and TF2B in case of eukaryotes. So it overlaps that region, which is the binding between these sigma region 4 and TF2B. This overlapping and binding raises the interesting possibility that displacing initiation factors may be part of the function of these elongation regulators. So it is suggested that it is the part of these elongation regulators to dissociate the initiation factors. This also suggests that regulating the rate of elongation is an ancient mechanism of regulating gene expression. There are some promoters in higher eukaryotes where the pre-initiation complex is recruited effectively, but polymerase remains paused just after initiating transcription. So all the factors are recruited and initiation complex is formed, and then polymerase remains paused. It stops the transcription after initiating transcription. Such promoters seem to be associated with genes poised to be expressed either rapidly or in a highly coordinated fashion. And their expression is regulated through recruitment by specific activators of the PTEF-B kinase, which then releases them from their pause, and then this pause is released. Yet another class of elongation factors is the so-called ELL family. These also bind to elongating polymerase and suppress transient pausing by the enzyme. So when these ELL factors bind to the elongating polymerase, it suppresses the pausing of this polymerase, that is stoppage of this polymerase enzyme. The first human ELL protein was originally identified as the product of a gene that undergoes translocations in acute myeloid leukemia. Another factor that does not affect initiation but stimulates elongation is TF2S. So TF2S is another elongation factor. This factor, like ELL, stimulates the overall rate of elongation by limiting the length of time that polymerase pauses when it encounters sequences that would otherwise tend to slow the enzyme's progress. So in the DNA there are some sequences which tend to slow down the polymerase speed. So this factor it overcomes this stoppage or this pauses of the polymerase. It is a feature of the polymerase that it does not transcribe through all sequences at a constant rate. So it varies the speed, and at some places, at some specific sequences, this polymerase pauses. Rather, it pauses periodically, sometimes for rather long periods before resuming transcription. So sometimes it pauses for very long periods. In the presence of TF2S, the length of time that polymerase pauses at any given site is reduced. TF2S also contributes to proofreading by polymerase. So it proofreads the newly synthesized RNA. TF2S stimulates an inherent RNase activity in polymerase, which is not a part of the active site of polymerase, allowing an alternative approach to removing misincorporated bases through local limited RNA degradation. So this is another function of this TF2S, which is proofreading by local degradation of misincorporated bases into the RNA molecule. This feature is comparable to the hydrolytic editing in the bacterial case stimulated by the GRE factors. So in bacteria, these GRE, they cause hydrolytic editing of the misincorporated bases in the RNA.